Hello everyone, and welcome back to the Beck Lab at Wichita State University. This is our second video on doing meiotic chromosome counts in flowering plants. And today we're actually going to do the prep. We're actually going to do the chromosome squash itself. We are going to be looking at material from this individual. This is buffalo grass, Chloe dactyloides. This is some material that my graduate student Jacob Hadel collected during the course of his master's thesis. We actually are going to be working with the fixed material from that specimen. And these preps involve, I guess, kind of three locations. The first place you work is on a glass dish like this, and this is working under the dissecting scope, where we will remove material, some of this fixed material, and on this glass dish we will look for floral material that's at the right stage. Specifically, we want anthers of a certain size and color, and we will try to isolate those anthers to the degree we can from the other floral material. Then we will move those anthers to a slide, and on that slide we'll work in two locations. The first place we'll transfer those anthers to is this location 1 towards the left of the slide. There we will remove any additional non-anther material that we can. We might even start breaking up those anthers a little bit at that location 1. And we'll be doing that work in the dilute acetocarmine stain. Then we will move that material, those very isolated anthers, to this position 2 on the slide, and we will be working in the concentrated acetocarmine stain at that point. And that's where we will fully macerate, fully tear up that tissue as much as we can at that location. The stain will be working into the tissue. And then we will remove as much of the large visible chunks of anther material as we can. And when that's done, we apply a little bit of Hoyer solution, put a cover slip over it, still on that location too. Then we try to push out some of the obvious bubbles that are underneath it. And then we apply pressure with our thumb to squash those stained anthers mixed with Hoyer's and acetocarmine stain under that cover slip. And then there's a little bit of cleanup involved, but basically that's it. Work on the glass plate, and we transfer the material to 1, transfer it to 2, tear it up, clean it up, and mash it under a cover slip there at location 2. All right, let's get going. What I've done here is removed some of the floral material that you saw just a second ago in that vial, and I've placed it on the glass dish. And now we're looking at it at relatively low magnification on a standard dissecting microscope. Buffalo grass is dioecious, so we're talking about male or female individuals. This happens to be, of course, a staminate inflorescence from a male individual. And if you remember your grass morphology, you know that an inflorescence is made up of spikelets, which are in turn made up of florets. Here's this inflorescence. There's several spikelets here. I've removed one of the spikelets. I've actually left the two glooms that subtend that spikelet down here. They didn't come along with it, so the glooms are still there on the spikelet, or on the inflorescence. Now here's the two florets that made up that spikelet. I can pull them apart from one another. This is often easier said than done. There they go. Each one of them, involves a palea and a lemma, which can be difficult to pull apart. What we want to do is we want to go inside that palea and lemma and pull out the anthers that are inside. Kind of tore that one in half, but there's 3 anthers from that floret. I'm going to get a little more ethanol in the mix here. That's part of the game here, is to add ethanol to the plate every once in a while to keep everything nice and moist. Opening up that second floret. I think you can already see that the anthers inside that second floret are considerably larger. They're yellower. which indicates that they are much farther along in microsporogenesis and probably contain what is essentially mature pollen. We don't want that. We want something a little bit before that, during the early stages of meiosis. And we would, in fact, rarely, if ever, and I guess never, want anthers that look like the ones on the right. We want ones that look like the ones on the left. We certainly don't want anything that's yellowed, like you see there. What I typically do when I start a new prep with an organism that I'm not familiar with is I'll look for the largest anthers that are not obviously yellowed. And I'll make a prep out of that largest non-yellowed size class and see if I'm still, it's possible those are even themselves way too far along in meiosis and microsporogenesis, and we're not going to use those. In that case, you can just make a second prep and a third prep and ratchet back on the size of anthers that you're incorporating. This is pretty straightforward to do in buffalo grass, or in many grasses for that matter, because the anthers are pretty easily isolated. In your organism, you may not be able to isolate the anthers themselves. You may have to isolate anthers along with associated floral parts and then transfer that to the slide instead of individual anthers. That's certainly true in Asteraceae, which is a group that I work with a lot. The anthers are simply too small and part of larger complex structures. You just can't isolate anthers themselves. So typically what I do is just try to remove, I find florets that are of a target size. And then I try to pull away pappus and immature cypseli from those, and then move forward with that floral material plus anthers and pistils in that case, and move those onto the slide and then go from there. But in the case of buffalo grass and in many organisms that you're working with, you can target the anthers directly. So what I'm going to do now is I'm going to cruise around and I'm going to find more anthers of this target size, the largest ones that are not yellowed, and I'm going to just kind of pile them up, and then when I've got enough, we'll transfer them to the slide. What you can't see here is that what I'm doing is I'm applying a bit of dilute acetocarmine stain to the left side of the slide, the position 1 if you recall. And I'm coming in and grabbing some of this material on I think it's the surface tension, perhaps is the term. I don't know what I'm looking for here. Will allow you to tease that up on the forceps. And I'm transferring it to the slide, which you can't see now, but you'll see in a moment. There we are. Now there's your drop of anthers in that dilute acetocarmine. And for funsies, I'm also going to add in a few goldenrod florets there as a teaching moment. Okay. So the whole point of this dilute acetocarmine step is to remove, it gives you one last chance to kind of remove anything that you don't want. In other words, anything that's not going to contribute to the prep. And in the case of buffalo grass, it's pretty trivial. Pretty much everything in here is potentially meiotic material. There's no accessory stuff that we want to remove. One thing that I often do at this step is perhaps, tear apart, go ahead and tear apart some of the larger anthers. You can just kind of cut them in half or rip them in half. You can do that kind of jazz if you want. And that's all fine and good, and that's what we would do with buffalo grass. However, the reason I brought in these goldenrod florets is to illustrate that this step can actually take quite a while and be very important in taxa where you can much less effectively isolate the anthers. So if we look there at our goldenrod floret that I'll try to focus, the image is not going to be nearly as good when we're dealing in these drops of stain. That's much better there. I don't want this immature cypsilla or the pappus, so I'm just going to kind of stab / cut, almost like a chopstick motion there. tease away some of the still attached pappus. So you can see now what I've got is something that is floral material. So tube, and then there's the anthers that are still left in there. So now I've got something, that item, which I'll transfer forward to the next step. At this step, whatever organism you're working on, you want to get rid of as much stuff as is practicable that's not anthers, and certainly anything such as pappus that is really tough and that is not going to break up in the next step, because anything that doesn't break up into small pieces in the next step, if you don't remove it at the end of that next step, it's going to keep you from getting that cover slip down really tightly on the glass. You're not going to get a good image, and you're not going to be able to focus clearly on any individual cells. So you can imagine that if this was a goldenrod sample, sometimes this step can take 30 minutes as I go floret by floret, removing the accessory materials that I don't want. Okay? So, but we don't want that goldenrod there. Rather, we're focusing on our pile of buffalo grass anthers, which are at the moment kind of slowly picking up a little bit of stain in this dilute stain. This dilute stain just gives you a chance to work on this and spend a lot of time with it without overstaining, without working in concentrated stain. And honestly, we could probably skip this step in buffalo grass and in other organisms like it, where you can directly isolate the anthers. Okay, I'm going to continue working here. All right, what we now have is a pile of anthers that I have torn in half, in thirds, smaller pieces, and now I'm ready to transfer this to the drop of concentrated stain. Now at this point, from this point forward, once you start working with the concentrated stain, this becomes time sensitive. So I usually take this point to make sure that I have a cover slip that is clean, and I've removed from the box. I'll hold it up to the light and make sure there's no lint on it or any kind of goo or streaks or whatever. So you got a nice clean cover slip that's ready. You've got you've got your Hoyer's vial open, your concentrated acetocarmine vial open. You've got your surface that you're actually going to press, flatten the material, do the squash, make sure that it's clean, that you've wiped off the surface to make sure there's no sand or grit there, that you're ready to work. And provided that you are ready to keep moving. So off camera, what I'm doing is I'm going into my now concentrated acetocarmine and getting a small drop, and I stress small. You want to start with a very small drop of concentrated acetocarmine, and I'm applying that to the right side of that slip off camera. Just put that little drop there, and now I'm going to go in and I'm going to grab this pile anthers, move them across the slide to the right to that drop of concentrated stain. And there we go. I'm going to pull the slide over, and now you see that drop. That's our drop of concentrated acetocarmine with our anthers in it. Well, the whole point of the working in the concentrated stain is to mash the bejesus out of the stuff, and then to go in and clean out the big pieces once you've done that. But we need to start mashing first. So you want to wait till that drop. The best time to do this mashing is when that drop of stain is almost gone. In other words, you don't want to be splashing around in a massive drop of stain. So I'm just continuing to mash these anthers. And again, your best work can be done when the drop is almost gone. You don't want this to dry out, but you want to work with it on the tacky side, as I would say. You can see as this material is almost drying out, not drying out, but is getting tacky, you can really do some damage to it. Now I'm going to add some more stain. So again, I'm going to my forceps, dipping into the concentrated stain and adding. You don't want to add too much. You just want to add a little bit here. That'll work. Add a little bit. I'm going to zoom out a little bit here. Do a little focusing here. Go in and work that stain in. All parts of the field. You want to try to, the edges are going to dry out. You're always going to be fighting against that. So you kind of want to do a little bit of work around the edges there. You're constantly going to be fighting that. I'm going to get back to it. As the little drop of stain is less bubbly, or if it's bigger, you want to be more careful. But as it dries out, as it gets closer and closer to tacky and dry, you can increase your kind of aggressiveness here. What I'm doing is I'm just holding that forceps at a pretty severe angle and using the last, I don't know, centimeter of it to hit that stuff. You don't want to go at too extreme an angle, because if you're almost perpendicular, I guess horizontal would be a better term, with the slide you can draw your stain down your probe, which you don't want to do. So you can see I'm really doing some damage here. And I want to add a little more stain. Back with the stain. Zoom out a little bit. Stain. You don't have to add all the stain that you pick up with your forceps. That'll often be way too much. You only want to add enough to kind of rehydrate your little working area there. Because if you add a big drop of stain, it's going to expand this working area, and you're going to have drying stain that you're going to be dealing with. It's just going to get an increasingly large area that's going to be difficult to manage. We're pretty close here. As you can see, what we've done is there's fewer and fewer identifiable anther pieces there, and what we're left with is mainly just kind of a blizzard of individual cells and small groups of cells that we can then mash and look at under the scope. we've pretty much reached the end of the point here where we're going to continue to trash this material. It's reached the grape jelly stage, more or less, as I call it. So what we want to do now is we want to go through a process where we remove as much of the large visible material from this as possible, and what we'll be left with is a slurry, kind of a solution of really small bits of tissue and individual cells that we can then look at. So at this point, what you can't see off camera is I have a little paper towel, just a regular lab paper towel next to me. Then I'm gonna start going in with my forceps, grabbing, using, teasing up bits of this tissue, and then just moving it over and wiping it on that paper towel. And just like the mashing, this becomes more effective as the bubble is smaller, but we'll just do an example here. So I teased up a little bit of material onto the forceps, really hardly any. So at this point, it's kind of a waiting game. I'm waiting for this bubble to decrease in size to the point where I can really grab a bunch of material in one fell swoop. While I'm waiting, though, I can kind of pull a lot of the chunks to the center. I can work on that edge a little bit to kind of keep it from getting super crusty. Again, without going crazy and expanding the size of this dot beyond the point which it would be manageable. All right, so here we go. I'm going to go in with these forceps. I'm going to use one to tease up a bunch of stuff on the other. You can maybe see that there's this big dark chunk of material there. I just kind of dumped it there again, but I'm going to grab it again, move to the paper towel. Do that a couple more times. Again, I don't want to run the risk of losing all of this. The field can't dry out, in other words. So I'm going to do that maybe once, twice more. Not pressing my luck. I'm pulling off this stuff that looks like grape jelly. Now I'm going to come back with more stain to keep the field hydrated. Good. Put that in around the edges. At this point, you can actually start to visualize some of those anther pieces that are still there. We don't want those. Those are going to keep making it impossible for us to get the cover slip flat enough here. What I'll do in this case, I'm just going to zoom out. Going in there, removing some of that grape jelly. Coming again, one time maybe. Quickly coming back with the stain. This is probably the most, if there is a nerve-wracking part of this prep, it would be this. Just because it's this game between keeping the field hydrated, but also working at that almost dry stage, tacky stage, where you can get a hold of some of the material. You can see if I just mix it all up, I've removed really a dramatic amount of that chunky stuff. You can't remove all of it. I don't want to leave you with that impression. You will drive yourself crazy trying to. But now I think you can see that we have removed the lion's share of that big stuff. Still some big anthers pieces in there. While we're waiting for that bubble to kind of get smaller, I can even go in and try to grab, so I just grabbed that one on its own. Now I'm getting to the stage where I can really do some work here, but again, I don't want to overdo it, so I'm going to maybe do one more pull here. That's good. Come back with the stain. Again, endeavoring to not make this field just become super big. Stain right there. If you drink a bunch of cups of coffee and then come in and try to do this, you'll notice the shake, and it will not be helpful. So, you don't want to lay off And I want to have just like 6 cups of coffee before you do this. I'm not encouraging anyone to drink no caffeine, but if you're going to do one of these preps, you might even notice I'm a little shaky here. I had too much coffee today. Nobody's perfect. One more stain. You see it's getting pretty dry there on that edge. I don't want that to happen. Come in, nice and worked in. I think I'm gonna hang it up. Just grab that little piece of anther. It's probably gonna be about as far as I go with this one. So I've added one kind of really large drop of stain here right at the very end. You can kind of see the array of larger anther pieces that I've just kind of given up and allowed to be there. I get all of them. Work that stain around the edges one last time. And the last thing I do under the scope, not really, I guess under the scope, is to go in and add a drop of Hoyers. And this drop is going to be, I might even try to post an image of what that drop looks like. Adding that right to the center, see that? And working that in, again with the side of the... And when you add the Hoyers, that'll give you kind of one last chance to, I guess, hydrate the field, if you like. And there we go. A mixture of a bunch of little individual cells. Kind of just appear as fuzz there. A few larger pieces. And concentrated acetocarmine and Hoyers. So now all we got to do is flatten it. Alright, so now we're back up in the lab and we're ready to actually do the squash itself. So here's our drop of Hoyers acetocarmine mix, and there's stained plant material in there. I'm going to apply cover slip. You should always have a clean, lint-free one ready. This one looks good. You want to gently lay it down over the field. And you should see the mixture slowly move towards the edges of the slip. This one looks pretty good. It shouldn't race to the edge of the slip. The fact that it kind of crawls to the edge of the slip is evidence of the fact that you've got the right mixture of Hoyers and acetocarmine. There's going to be some bubbles in this that are mainly they're going to form around the bits of anther and other tissue that you couldn't remove, and you want to push as many of those as is practicable out from under the slide. You don't want to push all of your material, all of this fluid out from under the slide, but you want to definitely move some of those bubbles. This will also help in that here around the very center of this slide is where most of this extra tissue is. And when we press on this, we're not going to be able to really flatten that area nearly as well as we will the areas near the two edges, kind of the east and west, if you like, edge of this slide. And that's where most of the counts are going to come from. I don't know, like a couple millimeters, frankly, on the east and west portions of this cover slip. So pressing some fluid towards those edges away from the center of the slip itself is going to push cells to that area where you're going to really be able to flatten them. So the way this works is you just you want to make sure that the slip is centered. Then you just want to put your fingers on the two edges closest to the two edges of the cover slip closest to the edge of the slide, and then just push those bubbles, just kind of chase them off to whichever edge is closest. This is one of the more whimsical parts of this prep, chasing bubbles. You won't be able to get them all. Some of them will just kind of bounce around. You won't be able to work with them. And there we go. Now we're ready to actually squash this thing. What you want to do is you want to get a standard lab paper towel, and using just under one fold. First of all, you want to make sure this area is very clean. You don't want any grit or sand there to break your slide. Tuck your slide under that paper towel, the first fold, and then kind of dab on it, and you'll see that you'll immediately see the outline of your cover slip show up because the stain will start to bleed through. We're going to press our fingers on two of the corners and then mash in the other corners that we're not pressing with our fingers. So I'm going to start with, let's say, corners 1 and 3, and then press in sections 2 and 4. So I'll start with section number 2 here. You want to press straight down with as much pressure as you can under control exert, straight down. Not side to side at all. You can use your body weight once you get kind of slick with it for about 15 seconds. Now I'll move to kind of corner 4, still holding on 1 and 3. Straight up and down. Now I'll move my fingers to corners 2 and 4. And maybe I'm pressing on section 3 now. I realize you can't see that all, but the diagram will make it clear. This thumb is covering, I don't know, approximately a little more than a quarter of the slide at this point. Straight up and down, as much pressure as you can bring to bear under control. We're going to finish with corner 1. About 15 seconds for each of these corners, let's say. And then finally what we want to do is, using both thumbs at one time, press on the kind of east and west portions of this cover slip. On the diagram, I guess the western one would be numbers 1 and 4, and the eastern numbers 2 and 3. So we get 2 thumbs down on those 2 sides. Again, because those edges, those very edges of the cover slip is where you're really going to get your counts. And I usually press for about 20 seconds. Then, this up a little bit. Remove your slide. And now what you want to do is seal the edges with Hoyer's solution. So you want to get a very small amount of Hoyer's solution and run it along the edges of that cover slip. Wipe in between applications. You don't want to use a lot because the Hoyer's as a clearing solution will tend to really clear those first few, first maybe millimeter or so next to this cover slip edge, and that's where we're going to get a lot of our counts. So we don't want to just dump a whole bead of Hoyer's along here that'll then clear the area that we're going to get a lot of our counts from. Just get the smallest amount you can really get on the end of that probe and just kind of run it along the edge of that cover slip. What you're doing is simply, that Hoyer's will dry tacky anyway, and it'll just seal the edge of that slide and keep any additional air from getting under the slide and expanding any bubbles that are underneath it. The last thing we want to do is we want to clean particularly, again, this first little bit on this eastern and western edges of this cover slip, because that's where most of our counts, this is where it's going to be the flattest, and that's where we're really going to get most of our counts. So this is very simple. You just want to get a new Chem wipe. Actually, you don't even need the probe. Just get a new Chem wipe, ball it up a little bit on the end, get a little ethanol, not too much, and then just run it along that extreme eastern and western edges there, just in case there's any stain or Hoyers that is now kind of bled over onto the top of the cover slip, that we're removing that. And so now what we have, and I'll actually switch to, because this is like a fishing show or a cooking show, I'll switch to the finished slide, the actual slide from this material that I did I guess back in December of 2015. This is a finished slide. So what we see here on this, add a little contrast, is this is the stained material, pressed stained squash material over here under the cover slip, and over here you see all the information. This is the collection number, the taxon, the date I did it, and then actually I know this thing ended up being a tetraploid, and so I usually write the count on there after I get it. But this is the finished product, and this is now ready to go look at underneath the light microscope. So this slide is ready. You can go look at it immediately, or you can set this aside. You can really store these anywhere. I usually like to store them flat anyway until I know that Hoyers is dry. Just keep the lint off of them, and you can wait several days to look at this, or you can look at it immediately. And that's pretty much it. Good luck, everyone.